okay? So oxidation reduction, if you're going to understand metabolism, you have to understand the movement of electrons. One molecule gives the electron up, that's oxidation. Another molecule accepts the electron, that's reduction. When you talk about metabolism and the transfer of energy, it's all about high energy and low energy electrons. It's all about electron transfer. So oxidation reduction. When we then talk about photosynthesis, please understand that this is a flow of energy. The ATP that is powering your cells right now, the energy that's in the ATP ultimately came from the sun, all right? So the energy from the sun is captured during photosynthesis and stored inside of macromolecules, food. That food is then oxidized for its high energy electrons. Those high energy electrons then go through a process called cellular respiration, which then transfers the energy from the sun, from the high energy electrons to the ATP. That then returns the electrons to their low position. They are replaced back inside of water, and the cycle's all over again. This is why water contains low energy electrons, right? Because the electrons get excited, then the energy is taken out to make ATP, and then the energy or the electrons are no longer excited. We first present this to you like this. We show you the chemistry based on glucose, because the carbon, hydrogen, oxygen process, the moving of the atoms is relatively straightforward, but you will find out on Thursday, if my internet connection works, that it's not really just glucose. It's a compound called glyceraldehyde 3-phosphate. We just call it G3P. It's a 3-carbon compound that is then used to make any macromolecule. For G3P, you can make amino acids, you can make lipids, and you can make nucleotides. Okay, so really this is G3P, but for the introduction. Okay, fine. Then to step back, don't forget that the plant is cycling carbon dioxide in and oxygen out during photosynthesis. This is where we teach children that, we breathe out carbon dioxide and they produce oxygen for us. That is not accurate, because then the plant turns right around and takes this oxygen back in for cell rest. Okay, so plants are green. Why are they green? They're green because they have an organelle on the inside called a chloroplast, literally in Latin, green bag. Then inside of the green bag, guess what? There's more green bags. So those individual green bags are called a thylakoid. We're going to talk about the thylakoid today. And that's where photosynthesis actually happens. Okay, photosynthesis has 2 main steps. One happens in the membrane of the thylakoid. We call that light dependent. And then there's a second step that happens out here in the fluid or the stroma of the thylakoid, and we call that light independent. So when you see this whole chloroplast here, don't forget this is an organelle. It's not a plant cell. This is one organelle that's inside of the plant cell. So when you take this, you then see this, right? So these little green bags here are those little green bags there. And we're actually going to talk about the membrane of that thylakoid today. So this is just a slightly different view. Processes are the same. Water in, oxygen out, right? Carbon dioxide in, macromolecules out, G3P. Okay, fine. All right, don't forget that the first step is dependent on light, right? Thus it is light dependent. The second step does not require light, thus it's light independent. The light dependent reactions do make a little bit of ATP, but not enough for the cell to live on, not even close, okay? The whole point of the ATP is to power the enzymes of the Calvin cycle over here. Cool, let's get rid of that. So we then need to zoom in on the light dependent reactions here, okay? So the light dependent reactions here are here. And there are 2 steps to the light dependent reactions. They are a complex of proteins called a photosystem. They're numbered after their order of discovery, not the order in the biochemical pathway, fine. So it's photosystem 2, then an electron transport chain, and then photosystem 1, right? The whole point of photosystem 2 is to create high energy electrons whose energy is then used to make ATP, right? Now, when biologists want to fake, when we want to fake specifics, here's what we do. We just draw an arrow. We just go, look, Electron transport chain, an ATP, poof, it just comes out, right? So as an educator, whenever you want to back away from details and specifics, you just draw an arrow and go, look, ATP is formed. So we will get into the chemistry here. It's not complicated, all right? We will get into these details. So photosystem 2, what's the whole point? To produce high energy electrons whose energy is then used to make ATP. That's what I want you to understand conceptually. Photosystem 2 makes high energy electrons whose energy is used to make ATP. And then this ATP powers the Calvin cycle, right? But now you're left with low energy electrons again. They aren't useful. So photosystem 1 re-excites these electrons, and these electrons are then captured here. These are the electrons that end up in our food. These are the electrons that put the energy into ATP that powers our cells, okay? This ATP powers the Calvin cycle. This is the energy that ultimately ends up inside of you and I and allows us to be alive. Comfortable?
Yes.
All right. Okay. So we're going to talk about a membrane. Now, if you just slap down this image, you know, students commonly will go, oh, look at that. And the context here is very, very important. So before we see that image, we'll be right back to you. I want you to realize where we are in the cell. So plant cells. Plant cells have a cell wall. Okay. And then inside of that cell wall, of course, around all cells, is a plasma membrane. So then this, boom, plasma membrane. Okay. Now there are dozens and dozens and dozens of different organelles here in the cytoplasm of the cell. Okay. Now, one organelle, and I'm going to draw it ridiculously big, okay, for detail. This would be then the chloroplast, originally named when they put cells under the microscope and saw green bags. They didn't know what they did, so they said, hey, let's use Latin and call them the green bag. It's literally what that means. Fine. So now inside of the chloroplast are even more bags like such. Okay, these are thylakoids. That's what we're talking about today, okay? Because embedded in the membrane of the thylakoid like such are the photosystems. That's actually the location of photosynthesis, like this. These are just studded with different proteins. That's where the photosystems are. They're right there. The photosystems actually sit. So this is photosystem 2 and photosystem. It's going to be a really thick one. Photosystem 1, right? As well as the electron transport chain. Okay. So when we show you this membrane with these proteins in it, this membrane is really this membrane here. This is the thylakoid membrane here inside of the chloroplast in the cytoplasm of a plant cell. Does that make sense?
Yes.
Cool. All right. So then to handle this image, just toss that over there. We need to be sure and put this into context. If I were in the mouse, it'd be right there. There we go. All right, so this is the thylakoid membrane here. Thylakoid membrane. Like such, okay? Which means then that there is a chloroplast membrane out here. Okay, so this is the chloroplast membrane, chloroplastic membrane, whatever. It's the membrane of the chloroplast. Well, if this is then the chloroplast here, Then what's here? And what's outside?
Cytoplasm.
That's the cytoplasm, okay? This is the cytoplasm of the plant cell. Okay, if this is the cytoplasm of the plant cell, then there's fluid flowing here, yes? What is this fluid out here?
Stroma.
Nice, nice. That's the stroma. It's just vocabulary. That's the one complaint about biology, and I get it. It's all the vocabulary, right? Okay, so let's go through the vocabulary. Okay, so comfortable that we're in the thylakoid, inside the chloroplast, in the cytoplasm of a plant cell. You good?
Yep.
All right. So you already know about 90% of the content. I just need to add a little content on this magic red arrow. Okay. So let's do orange and green. Is that what color I've been using for oxygen? Yep. So on the previous handouts, try to keep these colors to color coordinate them. I would definitely keep using the same color. So I used orange for oxygen and green for hydrogen. Fine. So here comes good old H2O. H2O, green for hydrogen. Can't make a mistake with crayon because to try to white out crayon is really hard. All right, so there's my water. The water is then ripped apart, okay? The water is ripped apart into what?
Low energy electrons.
Low energy electrons here. So there's your low energy electrons. And then what leaves here?
Oxygen.
Excellent, that's oxygen. Now, one of the items that I've been glossing over here is when the electrons are pulled out of the water, okay? Because in water, you have hydrogen and oxygen, okay? You take the oxygen, well, you plants take the oxygen and rip the oxygen out. So what are you left with? Hydrogen, okay? But you know hydrogen is really made of two things. This hydrogen is made of what two things?
Proton and electron.
Protons and electrons, okay? What the plant really does, all right, is yes, the plant grabs the electrons and the electrons are here. What have I not accounted for?
The protons.
The protons, okay? The protons are also just as important. The proton goes here. Now, this is where some students get confused because I wrote a capital H here and I wrote a capital H here, okay? Chemists write protons as H+. So please understand that is two atoms of hydrogen. So right there are two protons and two electrons. Whereas you have the two electrons here, and technically you have two protons here. That's a proton, not a hydrogen. So ultimately, since the electrons were ripped out of water, what process just happened to the water?
Pulses.
Yep. That's photolysis, and it is also oxidation. Cool. So you then have a yellow lightning bolt. Why? Why is there a yellow lightning bolt? What is that?
Light.
That is light. We'll even capitalize it because it's so important, okay? That light takes these low energy electrons here, right? Takes these low energy electrons and converts them to what?
Energy.
High energy electrons, okay? That happens inside of this complex of proteins here. This is a photosystem. Which photosystem? Two. If you could get on the naming committee, if we could please call that photosystem A, that would be awesome. To number them is ridiculous. All right, so now you have high energy electrons. Now, these high energy electrons, you guys ever play hot potato as children, where you'd sit in a ring of kids, people, right? And then you'd pass some object and they'd play music, or you'd be singing a song. And then when the music ends, whoever's holding the hot potato is then out and the ring of people gets a little bit smaller. It's called hot potato. Did you ever play this game?
Yeah.
Maybe? Okay, cool. Or you probably called it by a different name. Fine. These proteins here then play hot potato with the electrons. These high energy electrons are transferred from this protein to this protein to this protein, okay? So there is an electron transfer between these proteins. The electrons are transferred from here down to here, okay? So since these electrons are transported from here to here through a chain or series of proteins, this is then referred to as an electron transport chain. So all the electron transport chain does is it transports the high energy electrons from here to here to here and ultimately drops them off right there. Now, what happens here biochemically? As the electrons bond to these different proteins, they change shape. And as they change shape, they either have more or less affinity for the electrons, and then the electrons are transferred. Fine. But why are the electrons transferred? And what is the energy used for? Get a load of this. A proton is then captured from the stroma, okay? So this here, this proton from the stroma is then grabbed and forced. It's pumped into the thylakoid. So the more water that the plant absorbs, the more photolysis happens, the more protons that the plant can then pump into the thylakoid, okay? Until there is a very high concentration of protons inside of the thylakoid, okay? So much so that this protein here is called a proton pump. And that's a pretty high-tech name. It's called a proton pump because it pumps protons, okay? It pumps protons from the stroma into the thylakoid, right? So the protons that are here come from two places, okay? The protons that are here originally come from the water, or they're pumped in as a result of the electron transport chain. So the whole point of photosystem 2 is to create this high concentration of protons. What do you think about that? Comfortable?
Yeah.
Okay. See this as steps. I understand this is unfamiliar. I understand this is new. Okay. That doesn't mean it's difficult. These are steps. This is biochemistry, and it's this happens first, this happens second, this happens third, this happens fourth. Okay. Plant absorbs the water. The plant splits the water by photolysis, and oxygen is released. The hydrogen, technically, we say the water is oxidized, it's really the hydrogen. The hydrogen is then oxidized, right? You rip out those low energy electrons. The protons are kept in here. Those low energy electrons are excited by light inside of photosystem 2. Those high energy electrons are then passed or transported between these different proteins. As these electrons are transferred or transported between these different proteins, that causes this middle protein to change shape so dramatically that it grabs a proton from the stroma and forces that proton into the thylakoid. We call that pumping. So therefore, this is a proton pump. So the whole point of the electron transport chain is to pump protons, right? So now you're at the end of the electron transport chain. You now have two things. You have a low energy electron, and you have a ton of protons inside of the thylakoid. Do you want to go here with photosystem 2, or excuse me, photosystem 1, or do you want to go down here and make ATP? I have no preference. Which do you want to do first?
ATP.
ATP, you got it. Okay, so you go down here. There is a high concentration of protons in here. You have a low concentration of protons here. So these protons are then going to flow through this enzyme. As these protons flow through this enzyme from high concentration to low, we call that process chemiosmosis. This process of the protons right here moving from high concentration to low is chemiosmosis. My pen's working good. Chemiosmosis. Chemiosmosis is not pumping. It's flow. I cannot stress this enough. This is not pumping. This is flow. This is the flow of protons from high concentration to low. Okay? These protons flow across this enzyme. And you say, what enzyme? Okay? This is an enzyme called ATP synthase. Huh. What do you think ATP synthase does? Well, you read the enzyme backwards. This is ACE means enzyme. This is an enzyme that does what?
Synthesizes.
Synthesizes what?
ATP.
ATP. Thus the name ATP synthase. Okay? As protons flow through this enzyme changes shape. When the enzyme changes shape, it grabs a hold of an ADP and an inorganic phosphate and physically forces them together. This head, it turns and it ratchets and it opens and it closes, and I'll show you an animation later. There's a ton of conformational change here, but the concept is as protons flow through this enzyme uses that flow, uses the energy to change the shape of the enzyme, which allows it to bond ADP and P to make ATP. This is the ATP that powers the Calvin cycle, which I should put over here for you. Right? At which time, watch what the protons do. Because the proton has now returned out here to the stroma, right? This is back to the stroma. What does this proton then do?
Return
Return
to, the proton pump.
Yep, it just cycles. Put some highlighter on that too. And then the plant absorbs more water, absorbs more light, gets even more protons. It's just a cycle, right? Proton gets pumped in, proton flows out. Proton gets pumped in, proton flows out. It's just just merry-go-round is all it is. Okay, so since you have a high concentration of protons here and a low concentration of protons here, what you actually form is called a proton gradient. And you're going to see this again inside of the mitochondrion in a couple of weeks. That's a proton gradient, high concentration to low. So that's how it makes ATP. All right, what about those low energy electrons? What happens here?
Light is used to excite the electrons again.
It excites them again. That is then transferred to an enzyme here, which I think this looks like the little skulls they use for Dia de los Muertos. I've often say it looks like a tooth. So I always draw a little skull because I think it's cute. This enzyme captures these high energy electrons. Da-da, da-da. What goes here?
NADP.
Nice. NAP to make?
NADPH.
Very nice. And so NADPH then is now carrying the high energy electrons here that end up in our food, meaning that this is which enzyme right here?
NADP.
Very nice. NADP reductase, because that is the enzyme that reduces NADP into NADPH. So now at the end of the light-dependent reaction, what do you have? You have ATP to power the enzymes of the Calvin cycle, and have high energy electrons that you're going to bond, or the plant, to carbon dioxide. Holy smokes, that's the real deal. We're not playing there, right? There's 3 processes that are a big deal in biology, okay? The time that I transcription, translation, the first show you the central dogma, you go, whoa, and that washes over you. You go, okay. And then now you sit there and go, transcription, translation, big deal. This is the same experience, right? You see the light-dependent reactions and it washes over you. Wow, okay? That's a lot of detail. It's not impossible to learn because there's steps. It's just step, step, step. What happens next? What happens next? What happens next? And there's terms that you haven't heard of before. You're not sure what an electron transport chain is. So think about it. Oh, that's a chain. That's a series of proteins that transport electrons. Why? To pump protons. Why? To create a proton gradient. Why? So chemiosmosis can happen. Why? To make ATP. Why? To power the Calvin cycle. It's just, this is toddler. This is the classic toddler experience. Okay. Hey, so what do you think we're talking about on Friday if the internet cooperates?
The Calvin cycle.
Calvin cycle. Okay. And we'll take high energy electrons, carbon dioxide, and we'll bond them and we'll make repeat. That's all there is to it.
Mr. Manro,
Yes, ma'am.
is this like a specific question on core, or are we just reviewing all the images?
It's a great question. It is. I don't go through the questions yet, okay? Because these questions are ridiculously full of vocabulary. Okay, so if we go from we can read these questions and you read them and you go, that means nothing to me. Okay. So what I do first is I talk about the processes and I give you background, I give you structure, I give you context, okay? Then we go back to the questions and it says, diagram and discuss how high energy electrons from photosystem 2 are used to generate a proton gradient. Your discussion should include how ATP synthase in thylakoids generate ATP using the proton gradient. If you were to have read this question an hour ago, you're like, what are they talking about? Okay, this question here is what we just talked about here with the electron transport chain and the proton gradient. So we start with the diagrams first and we go back to the questions second because we have to go through all of these images in order for the vocabulary here to make sense.
Okay, thank you.
Okay. On photosynthesis and cell resp, you have to start with the diagrams first. You just have to, because it's crazy to start with the questions, because otherwise thinking it's just a big wave of vocabulary in the questions and you don't pick it up. All righty. I think that's more than enough content for one day. Do you agree? Yeah, that's a lot. Okay, so we'll get together. Go ahead and form your groups of 4s and 5s, and if you could type the names of the people in those groups, that would be great. And if you have any questions on the solved questions, let me know, and I'm happy to help.